Hello all, and welcome to the ZOOMinar and the molecular basis of proteinopathies. We have two excellent speakers today who are experts on protein aggregation, Dr. Roland Riek from ETH Zurich, and Dr. Anant Paravastu from Georgia Tech. I also would like to welcome another guest, Dr. Sebastian Hiller, who is also an expert on protein aggregation in NMR spectroscopy. Sebastian will introduce the first speaker, Roland Riek, and moderate the presentation. Sebastian, take it away.
Yeah, thanks a lot, Rams. Thanks a lot for having me here. And it's a great pleasure for me to host and introduce Roland Riek, Professor Roland Riek from ETH Zurich. I actually have a very long relationship with Roland, and this dates nearly back to 1998 when Roland did his PhD at ETH Zurich in the group of Kurt Wüthrich. And me and Roland just met a few years afterwards when I did my PhD, also then later in the same group. So Roland had a very successful PhD, which was maybe that the highlight of it was the structure determination of the prion protein from mouse at the time, which was highly innovative, could not be crystallized. There was no cryo-EM at the time, and the determination of that structure with NMR was a big feat and worldwide recognized. And then Roland moved on. He then next went to the Salk Institute. That was in 2001. First as an assistant professor, and then in 2006 as an associate professor. Salk Institute is in La Jolla in California. And then in 2007, he actually returned to Switzerland. He was hired back to ETH Zurich as a full professor to the Laboratory of Physical Chemistry in Zurich. And he's there ever since and doing highly successful research, a lot of publications, very impactful studies, as you will certainly see later in his talk. Roland received a number of prizes, and I want to mention only two of them: the Günter Laukin Award, which is probably well known to all of you, in 1999, and he also got an ETH medal for the best PhD thesis, which is something that means particular to me something. It's a good recognition of a fantastic work. So Roland, thanks a lot for being here today. Thanks a lot for speaking to us, and me, and I'm sure everybody here is looking forward very much to your talk.
Thank you very much, Sebastian, for this very kind introduction. And of course, you mentioned already that we collaborate, and I will show a little bit of this work also today. It's a great pleasure. Thanks, Rams, for organizing this. I really like this Zoom interviews and actually conferences and seminars, because they are also recorded, so one can watch it whenever one likes to watch it. And I would like to take the chance today to talk to you about amyloids from a structural point of view, and show three examples. Oops, what happens now? Okay. I will show you a hormone amyloid system that is a functional amyloid, and then relate that to a disease-associated amyloid system, alpha-synuclein, related to Parkinson's disease. And at the end, would like to also exploit the structure of an amyloid in a so-called amyloid world hypothesis. We and others stated recently that states that peptide amyloids may have played an important role in the evolution of life. And what's important is to understand that an amyloid is a unique protein structure. We call that the cross-beta structure. In 1930s, described the first time by Asbury. And it has these repetitive structural elements where we have intermolecular beta strands that are 4.7 angstrom apart of each other and are repetitive in nature. So you need several molecules that come together to fold this entity. It's a very stable entity. It can also be sensitive to the environment, as we shall see. And when they are different structures of an amyloid fiber, we call them polymorphisms. So let's start with in life with a functional amyloid. So nature uses this amyloid fold in many different areas. I wrote recently with Daniel Olsen together a review, and we listed, and I show you just now here is site by page systems in the entire animal kingdom that are amyloids of functional nature. And I'm positive there are more and more coming. And I would like to explain you one of the systems we studied over the last years intensively, and it started with Samir Marchi's work when he was still at the Salk Institute. I show you here a secretory granule where it is depicted in this cartoon that a hormone, we will talk later about beta endorphin hormone, forms an amyloid. The amyloid is an inert storage. It's the highest dense packing possible of a structure based on argumentation from David Eisenberg. And that amyloid is probably inside this electron dense material. It's a membrane. You may see here this membrane entity surrounding this electron dense material of the secretory granule purified from rat pituitary. And when we look at this and study these secretory granules spectroscopically by a dye that measures amyloids, we see that they are THT positive. They show congruent birefringence. They bind to an antibody in a dot plot that is somewhat, not exactly true, but somewhat specific for amyloid fibrils. It's the OC antibody from Charlie Glabe. And we see a 4.7 angstrom band in fiber diffraction, telling us indeed we have this beta sheet, this cross beta sheet motif, and the second layer in the back of it. And this band here is from the lipids that are still in the secretory granules, purified from rat pituitary. So we are a structural biology lab, so we were happy to get rid of the material and concentrate on structural work. We started that work before the cryo-EM revolution. We got beautiful cryo-EM pictures at that time, but decided to do solid-state NMR with it. And that's the three-dimensional structure of beta-endorphin hormone in its amyloid state. Beta-endorphin is a hormone that usually kicks in about kilometer 10. Of course, that depends on personal qualities, at about kilometer 10 in a marathon to suppress pain. But it is stored in secretory granule in this fibril structure. So we have intermolecular beta sheets. So each layer is a molecule. Here is molecule 1, here's molecule 2, here's molecule 3, and it will go on like that for hundreds of repetitive elements. And so we have this intermolecular beta sheets, so a highly stable structure. That structure we determined at around pH 5.5, which is the secretory granular element pH. Now, if you look from the top, you see these three beta sheets that come together, and the core I depict now in the next slide is composed of some hydrophobic areas like this phenolene in here and this phenolene in here, but it has also a hydrophilic core with a glutamate. This is a glutamate here at position 8, but solid-state NMR can tell you that this glutamate is protonated. It's not negatively charged, and so it can reside inside the fibril core. Now, how that comes, right? So why should that be such an amyloid formation, and why should there be a glutamate that is protonated? Of course, these amyloids should fall apart when the secretory granules are secreted and go into the blood, which is a pH 7.5. In the secretory granule, we have around pH 5.5. In addition, in secretory granule, we have phosphate as a buffering system, which is not existing in the blood. So we looked at in vitro at this change of buffer condition by diluting it. Of course, a dilution happens as well by changing the pH from a phosphate environment to a tris environment, therefore also changing the buffer. And then the measure of T-flavin T, which is the amount of, is a measure of how much amyloid fibrils you have, you can see drops strongly because the fibrils fall apart. And they need to fall apart because they need to disassemble. So there's a function amyloid that is sensitive to the buffer condition. and the pH. And now, of course, it is evident to think that this glutamate 8 is the pH sensor of these fibrils. And when the pH increases, the glutamate gets negatively charged, and then the fibrils fall apart. Now, to get insights into that, we made mutants, getting rid of the charge, E8 to glutamine or E8 to leucine. And you can see now that in comparison to the wild type that is sensitive to the pH in the solution, the two mutants are much less sensitive. And that's because this glutamate is now getting deprotonated and gets negatively charged. But we measured with solid state NMR, and actually the glutamate inside the fibril are still protonated. Only at the edge, water is able to access and deprotonate the glutamate. And therefore, the effect is not so extreme, but that may also be a very highly organized way to dissolve from the fibril and in an organized manner the hormones, so that you have a slow release of hormones in the blood. But this is a little bit of speculation. So from this point of view, I hope I could show you that this amyloid system here in the secretory granule is an inert storage of a hormone amyloid, but then that falls apart when it is necessary to fall apart. So a highly functional amyloid system. That changes entirely when we go to a amyloid system associated with disease. In this case, it's Parkinson's disease, and we talk about alpha-synuclein. And everything gets much more complicated, as we shall see. And that's also probably true because it is not evolutionarily optimized to do so. Together with Sebastian, we started to work on alpha-synuclein in cells. So we make N15 labeled alpha-synuclein, electroporate that into mammalian cells, as described by Phil and others. And we get about 4 micromolar of synuclein into the cell. We can measure then NMR spectra and try to understand how synuclein looks like. And I would like to stress that something I missed for a long time, that synuclein is actually, although an intrinsically disordered protein, so you would expect that it's probably something like that with an N terminus here and the C terminus here, but that's actually not really true. It looks like that the N terminus of synuclein comes close to the C terminus because of the charge and some aromatics. With the N terminus is highly positive charge, the C terminus is highly negatively charged. And that's highlighted by a figure here from Phil Zelenko. That is even found in cells, not only in vitro, as described first by Zweckstetter and others. So although it is moving around a lot, it somehow is coming close by, and then the NAC region, this non-amyloid component, that this NAC region is, which is important for aggregation of synuclein, is then a little bit protected, and synuclein actually is a very good inhibitor of itself to aggregate. So that's the summary of from others, and we started then together with Sebastian, work on alpha-synuclein cells. And one means the signal we find in vitro, and when we have less signal in the cells, like the first 20 residues here, that means we have transient interaction of let's say that would be the N terminus of synuclein. Transient interaction, that means interaction in the microsecond time range, something like that, with a protein partner inside the cell. And also around tyrosine 39, there is interaction and some unknown interaction, although we have an idea, but still didn't do the experiment, that the C terminus may happen. So to get insights what actually interacts in the cell, there are many possibilities, right? So we made the following construct. We made a delta variant of synuclein where the first 10 residues are missing of synuclein, and did a pull down and a comparative pull down with full length synuclein, and look which proteins are now less good by the truncated variant pull down. At 1, that means they have the same amount of binding. At 0.5, the internal truncated one binds less. And what you can maybe see here, there's a lot of HSP chaperones that come down. Now, Sebastian is an expert in chaperones, and so he has all these proteins, and he then in vitro showed that indeed, for example, if you take HSP90, the N-terminus, and around tyrosine 39, we see a drop of the signals. That means a transient interaction of synuclein with HSP90. We then said, okay, if that is, if we have some evidence now in vitro and based on this mass spec pull down, can we do that also with in-cell NMR? And we took a cell which has HSP70 there, and then we measured an HSP70 alpha-synuclein electroporated and HSP70 depleted cells, and we don't see any change, as you can see. So that's now, the reference is now to the cell, which is just the normal hex cell line. And then we added in addition drugs that should interfere with HSP activity, and then we see signal coming back because it doesn't interact anymore. That signal that comes back is like the negative that we saw before. So there is now no interaction with the HSP70 or 90 anymore. And now after 24 hours, we see a new signature, a transient interaction of the first 100 residue with something else. And that had been shown before that this is probably unlikely to be membranes. So that synuclein now usually regulated by binding to chaperones, if that interaction here is interfered with, starts to interact more with membranes. And that we did an in vitro experiment with that, where we see now indeed here we have large luminocellular vesicles, that the signal decays of the first 100 residue here of alpha-synuclein, because the first 100 residues of synuclein binds transiently. They bind off and on and off all the time, that bind transiently. But when you add chaperones, you can see suddenly the chaperone binding motif comes, and when you add more and more chaperone, you see more and more chaperone, but less binding to the membrane, because the two are fighting against each other, are competitive with each other. So when we do look at this 24-hour later experiments, we find also aggregates of alpha-synuclein, and there we find synuclein colocalized with mitochondria. And that's interesting because it has been also suggested that Parkinson's disease is a mitochondrial disease. And what we then did is, this is early data here, we have alpha-synuclein and we added to a lysed mitochondria. And we did an MTSL experiment, that's for the experts, where we kill the NMR signal that is close in space to position 90 here. So we have a paramagnetic probe that kills our NMR signal. So if you have the synuclein folding like that, and we have here this killer, so in this area here, surrounding this cysteine 91 position, we had to make a mutant for that. All the signals that are in vicinity especially are getting killed. But if synuclein would be unfolded, they would not be killed. And that's actually these experiments that Zweckstetter already showed, that actually the N-terminus is still sometimes close to position 90 because there is a little bit, or here is a little bit of signal loss. But when we add synuclein to mitochondria, then we get more signals. So that means synuclein is getting unfolded when it goes to mitochondria. So it is more like this conformation. Here, it's now, of course, now here in the cartoon depicted extreme, but it is a little bit less folded. And of course, we are now trying to understand what synuclein is doing that it gets unfolded, because then it may start to aggregate. And we are investigating that. Another thing we looked at is when synuclein may actually also transiently interact with its fibrils. So if you have the fibril of synuclein, it has been proposed by Dobson and Knowles that synuclein binds to the surface of a fibril somehow and starts to aggregate, the so-called secondary nucleation. And we investigated that also. And that's work by Cedric Eichmann in my group, and he's now with Phil Zelenko, but he is the senior author of this work. And so what we did is now we have fibrils, which we add to synuclein, and then you see that the first 15 residues are losing signal because the first 15 residues, or actually more, it's more like the first 30 residues, are actually interacting with the fibrils. But when we add a C-terminal truncated fibril, we don't see any more interaction. And that indicates now when we have a fibril that we have a flexible tail at its C-terminus of the fibril, and that is negatively charged all the time. And that now the positively charged N-terminus of the monomeric synuclein comes close by. Because the N-terminus is positively charged. The story is a little more complex because it's also about hydrophobics. But then alpha-synuclein unfolds and is ready to aggregate because the neck region here in the middle is now not protected anymore. I would call it, but I don't know whether that's a good wording, that synuclein is actually self-chaperoning, but Sebastian may say something about that. To show that actually synuclein unfolds, we again measure the same experiment before. We have this empty cell at position 90 that kills the signal in close proximity. And when you add fibrils, you get a little bit more signal back because synuclein is now less folded and is actually also by EPR, we measured the distance between 20 and 35. This distance we measured. And this distance, when you add the fibrils, change in average from 2 nanometers to around 3 nanometers, and that we did together with the Jeschke group at ETH. So what I would like to say is it's a highly complex system, right? We have a synuclein that is intrinsically disordered, but not quite, that it can interact with its N and C terminus. Then it can interact with its fibrils. Then in cells, it may interact with the chaperones or the membranes. But there is another story I would like to highlight you, the surface of a fibril, because we have many negative charges of this flexible tail of the fibril that actually there are a lot of protons attracted to this surface, like on a membrane with its negative charged lipids, and the pH on the surface of the fibril is about 1.5 unit lower than the bulk pH. And that we can also measure by solid state NMR. I don't want to go inside, but we did that with Thomas Wiegand. Okay, so what is the structure of the fibril? There's a next chameleon-like character that synuclein we have to talk about, because what we find is that depending on the buffer condition, I just show you now two polymorphs. You get very different polymorph structures. So I hope you enjoyed that morph that I got from Henning Stahlberg, who solved the cryo-EM structure, and we helped a little bit. So there's a huge conformational change of the alpha-synuclein fibrils, either and only caused by the phosphate in the system. And why could that be the case? Now, look at that. We have here several lysine residues that face each other, and you have an electron density, and we think that's the phosphate, and that stabilizes this conformation. If you don't have phosphate, then of course an entirely different arrangement needs to be, because the three positive lysine residues will not like to be close to each other, and they are actually in a different polymorph very far away. So the environment is like a chameleon. The environment of synuclein is causing a different conformation. So summarizing the synuclein story, we have an intrinsically disordered protein, but not quite. Then synuclein is regulated probably with chaperones in a transient fashion. If that interaction is interfered with, it may start to bind more to fibrils, may get localized into mitochondria, and in mitochondria it is less structured and opens up and may be prone for aggregation. It may also find its fibrils on the surface of the fibrils. It may be able to go on open up and aggregation is induced. And the fiber conformation depends on the buffer condition. On top of that, synuclein can make liquid-liquid phase separation that has been discovered by Salman Machi, and this is his work. So finally, I would like to come to the origin of life story, just a few slides. I would like to show you the rationale. Why could be peptide amyloids the origin of life? Because simple short sequences can actually form a tertiary, even a quaternary structure, which is very stable under harsh conditions, and many other points. But of course, we should do experiments. We should start with prebiotic conditions and try to make amyloid fibrils. And that's what we did. First, we looked at data that people collected on meteorites, where one finds mainly glycine, alanine, aspartate, and valine. We find also in the Miller-Urey soup, one finds these amino acids. Then we added volcanic gas, COS, carbonyl sulfide, which short peptides have been able to be made by Orgel and Gadiri. We explored that in more details to get longer peptides, and indeed just adding some volcanic gas to a mixture of alanine or valine or more complicated mixtures, we find amyloid fibrils. We have more data just than the EM pictures, but I just would like to highlight that to you. And then it's about replication. Can now these amyloids chemically replicate, not in terms of conformation, but chemically? Can they make themselves chemically? And we started a rational approach like in the DNA. We have a template and we have a primer, a short primer with alternative hydrophobic and positive charges. And the template has negative charge every second amino acid and the hydrophobic. And when they come together, and adding volcanic gas, then the question is which amino acids are added here at this position, and the comparison is just the primer alone. And I show you here now HPLC. We have a lot of data, just show you one. We start at this peptide here, and for example, in the supernatant where only there is no amyloid, the wrong arginine is added. But while in the amyloid, it's very little is added. But the right addition, having here a phenylalanine and then an arginine and then a phenylalanine, so adding 3 amino acids to the peptide, you find only in the amyloid, right? In the black, where you have only the substrate or the primer, I'm sorry, only the primer, then you don't get it. So if there are amino acids around that are activated by volcanic gas, then an amyloid can chemically replicate its peptide structure. But of course, we have other players nowadays in life, RNA and membrane. And I just would like to show you one example, how we bring these worlds together. So we take a membrane, it could be a fatty acid vesicle or a lipid vesicle. For example, we added a small peptide, and then outside we have activated valine in red and a lot of aspartate. And the valine and aspartate can make peptides, but since there are a lot of negative charge, they form not amyloids. If you add a lot of aspartate, as shown here, outside you don't find any aspartate, but the valine goes through while the aspartate doesn't go through the membrane. So the membrane is a barrier or a selection. And then you find here inside amyloids. I just blow that up. Here you find amyloids. We find even amyloids here, indicative of maybe amyloids can perturb the vesicle structure and therefore may do something with daughter cells. That's something we need to investigate. And the last thing I would like to talk about is RNA binding. And I'd like to show you that because it is a nice example of the repetitive structure of the amyloid. Every 4.7 angstrom, we have an N-terminus which is positively charged in the amyloid. And in the RNA, of course, we have the opposite with the phosphates, not in the same distance, but we have again phosphates at every couple of angstroms. And even if the individual interaction is very weak, maybe in the millimolar range, we expect actually cooperativity of this interaction. And we studied that. I show you just once data point here. So we have, I can now not see that, what I'm actually trying to show. Okay. So we have D nucleotides here with one phosphate. They do not bind, the RNA doesn't bind to amyloids. We have different amyloid systems here. Then the 3 nucleotide doesn't bind, with 2 phosphates also doesn't bind, but the tetranucleotide with 3 phosphates binds rather well to amyloids. And the 3 nucleotide with 3 phosphates binds also rather well. And so it is an amazing correlation for us between the length of a codon and the amyloid binding capacity. And if you know that we have GXG as the codons of the most 4 primitive amino acids, alanine, glycine, valine, and glutamate, and we need only actually 2 nucleotides in the beginning, where there were only 10 to 15 amino acids available. The length of 3 may be due to the binding to the amyloid. Okay, with that, I would like to finish. I'd like to thank for your attention. I showed you 3 systems: a functional amyloid system, a disease-associated amyloid system, and peptide amyloids. And I just hope that you got the concept, how powerful that amyloid structure is because of its replicative structure at sub-nanometer scale. And with that, I would like to thank the people. The Alpha-Synuclein story with chaperones is, as I mentioned, a collaboration with Sebastian Hiller, and Björn Burman worked on it, Juan Guerres in my group mainly. Cedric Eichmann's work that has been performed mainly by Pratibha and Diman, I showed on alpha-synuclein nucleation. The Cryo structure work I showed is, of course, a collaboration with Henning Stahlberg, who is now at EPFL. I'm sorry, it's still this wrong address. And the Origin of Life work is mainly done by Jason in my group. And Beat Meyer did help us with the solid state NMR spectra of beta endorphin. I would like to thank you for your attention.
Great, thanks a lot, Roland. Fantastic talk. Thank you very much. I don't know where I can clap, and we see maybe the audience clapping. No, not necessarily. Yeah, there's 2 more. Rams is also applauding. Thank you so much. I will be glad now to read questions that have been put in the Q&A, and everybody listening, please also enter your questions or comments into this section. I would like to start with a comment from Yifat Miller. He says, excellent talk, Roland. Do you think that also in alpha-synuclein oligomers, N-termini of alpha-synuclein bind C-termini of alpha-synuclein?
It could well be, but that doesn't need to be. We don't know. I mean, if you look at Simeone's work, I don't think that's very clear. I think in a phase transition, in liquid-liquid droplet, that's what happens. The intermolecular interaction goes into an intramolecular interaction.
Good. Anup Arunagiri says, nice talk, Roland. 3 questions for you. First question, what is the significance of chaperone binding to alpha-synuclein, which is an IDP?
Yeah, so I think that's a concept or a hypothesis that Sebastian and the other co-authors and we entirely try to allude on, that actually interaction of an IDP to a chaperone is a regulatory mechanism, how a cell can regulate IDPs by transit interaction. And this is, of course, a hypothesis at this time point, but we need to look into this, right, Sebastian?
Yes, of course, we do. This is also actually, as an anonymous attendee says, please don't confuse biologists. Synuclein is an IDP. Yes, yes, it is. The second question.
Yeah, okay. Yeah, it's an IDP, an intrinsic disorder protein with some residual long-range interactions.
The second question from Anup is, does the chaperone binding affect alpha-synuclein aggregation in cells?
Yeah, that's what we showed when we downregulate chaperone activity, we find synuclein in an aggregated state.
The third question is, are there any functional roles alpha-synuclein could have in mitochondria?
We don't know the function of synuclein, right? I mean, it looks like that there are some indications that it is important in vesicle transport, but we don't know the precise function. Now, we figured that something very strange, that when alpha-synuclein goes into mitochondria, that AK2, adenylate kinase 2, is a binding partner of synuclein, and it actually enhances the enzymatic activity of AK2. We don't know that's in vitro experiments, whether that's relevant, but it could be, right? Yeah.
Maybe a quick question to Rams. Shall we go through all the questions here, or for interest of time, first go to the next speaker?
You can take a few more questions, and definitely we have time. 5 more.
Okay, fantastic. There's really good questions in the chat. So the next question comes from Sayed Hadi Ali Janwan. Thanks a lot for the nice presentation, Roland. What is the differences between random coil and IDP?
I think one is a structural definition, the other one is a biological definition. To me, they are the same. Now, I mean, random coil is a superposition of all the possible configuration that the protein can go into, and it is doing that in exchange in a very short time scale. And an intrinsically disordered protein is to me something similar. Sebastian, please correct me, or Rams, if you.
Well, the random coil is the typical configuration state of the IDP.
Yeah.
Okay, great. Jeff Brander asks, do you have any idea of whether monomeric alpha-synuclein binds to the end of fibers or to the lateral surface? For example, by saturation transfer difference, NMR of sonicated versus unsonicated fibers.
So we have data, in particular Thomas Knowles, and we repeated this experiment, that you can look in kinetic experiment, whether it's at the end of the fibril or at the side surface of the fibril, because it is a different mass dependency. You have very little ends and you have large side surface. And actually it is binding to the side based on kinetic interpretation of the kinetic data.
Very good, yeah. Next question from Jeff Brander. Is there any evidence of enzyme-like activity from amyloids in the amyloid world hypothesis?
Yeah, so we and others have shown that when you have peptide amyloids, for example, with 2 histidines, and you have a metal ion like copper, you can have enzymatic activities in terms of hydrolysis, and it's a broad field that we are not following up anymore, but there are many amyloid fibrils. They are called also, in terms of mass, the most efficient proteins or peptides in terms of catalysis. But of course they are bad because they have a large surface area without grooves where the catalytic rate could be increased significantly, but per mass they're rather effective.
Okay, I will stay at alpha-synuclein and take a question from Giuseppe Melaccini. Great talk, Roland. You mentioned that the alpha-synuclein fibril monomer interaction is more complicated than just electrostatics. Could you comment further, please?
Yeah, so what we added is we added a lot of salt, and then we saw still interaction. And then we did also mutagenesis on getting rid of some aromatics. And so there is also aromatic interaction, long-range interaction between the N-terminus, also tyrosine 39, but phenylalanine 4, and the C-terminal tyrosines of synuclein.
Thank you. And one question from Martin Muskol. I was wondering about the rate of amyloid dissociation for beta endorphin. The 1-hour dissociation rate seems very slow for a hormone circulating in the bloodstream.
That's a good question. It is a few minutes. I mean, in terms of, if you would like to show the half decay, but we didn't dilute it as much, right? We diluted it only by a factor of 20, and if you dilute it into the blood, it's much higher dilution. On top of that, secretory granules are very tiny and small. And our fibers are rather big, and we have indication that they actually start at the end to dissolve. So the amount of ends matters. And so we think that is the reason why we have a slower decay. But in cell experiments, actually, where you have secretory granules and you have secreted secretory granules, they take also a few minutes to dissolve in some cell biology experiments. So maybe it's even good to have a slow release. I mean, actually, there are even drugs out there against prostate cancer treatment that use GnRH analogs that form amyloids that they release over weeks, over 100 days, amyloids, that injection into the subcutaneous layer of the body, you get amyloids, and then they release over hundreds of days hormone. And so you need only one injection per 100 days, not every day an injection.
Okay, we have one question from David Eisenberg. Roland, can you expand on your explanation of the binding of RNA phosphates to the 4.7 angstrom spacing of molecules in amyloid fibrils? The separations differ.
Yeah, so this is we are now going for the structure, right? We need a trinucleotide binding to the amyloid to understand, because the phosphate distance in an RNA is not matching with the amyloid distance. So we have, of course, two beta sheets. Is it now going from one beta sheet in the cross beta sheet motif to the next one or not? Is it actually needing the first and the third phosphate, or the middle as well? Is it actually the ribose? Is it important, the backbone? It's actually what we see at the moment. It's just the distance of the phosphate is important. We can get rid of the ribose and just make a C letter. But the 5' 3' is important. If you go 5', 2' on the RNA, then we lose binding. It looks also, so we are doing a lot of chemistry and understanding interaction, but at the end we need the structure, David. You're probably agreeing with us to really understand, because we need to know the specificity between the amyloid and the codon, and then only then we can, the codon in terms of trinucleotide, we probably can decipher the genetic code, because we don't talk about specificity yet.
And the final question here is a bit maybe philosophical also, but it's a very interesting question. From Grace Royapa, does the protein concentration increase as we age? Any thoughts, Roland?
I mean, what I think is rather that the ATP machinery, that the chaperones, that you have less ATP in a cell, that the control machinery is getting slower, and that's why aggregation starts or may start to happen. From a physical chemical point of view, of course, aggregation is entropy increase, so that you may say, okay, yes, we get more and more aggregation when we age, and therefore also higher protein concentration. Aging is for me an interesting problem that I don't understand. Some people say it's programmed, like others say that's just not evolutionary selected against. So it will be interesting to see how that comes along. But protein concentration in terms of aggregation, I think they increase when you age, but this is a hypothesis.
Thank you very much, Roland. Thank you so much.
Thank you, Roland.
Questions.
Thank you very much for the fantastic talk, Roland. Thanks, Sebastian. So next speaker is Dr. Anant Paravastu from Georgia Tech University. Anand completed his bachelor's degree from MIT and then went on to do a PhD degree in chemical engineering from UC Berkeley. There he worked with Dr. Jeffrey Reimer. Then he joined NIH to work with Robert Tickord in the chemical physics laboratory as a postdoc fellow, where he worked on developing and applying solid-state NMR spectroscopy to study amyloid fibers, particularly amyloid-β, He then joined as an assistant professor at Florida State University and the Magnet Lab, National High Field Magnet Lab. And then he moved to Georgia Tech, where he is now associate professor, Johnson faculty fellow, and Thomas A. Solak faculty fellow as well. His current research focuses on protein misfolding and amyloid aggregation related to Alzheimer's disease. So with that, Anant, it's all yours.
Thank you very much, Rams. Thank you for the kind invitation. Thank you, everyone, for listening. And thank you to Roland for a wonderful talk. In fact, you already illustrated a lot of concepts that I think are very important for my talk. So I do solid state NMR. My talk will be far less far-reaching and philosophical. I want to address a very simple question, almost a geometric question. So this is a small segment of Alzheimer's beta amyloid, and it is arranged into an in-register parallel beta sheet. So that is a very common structure for an amyloid. In fact, I believe all the structures in Roland's talk had this motif. If I color code hydrophobic versus charged residues, you might notice that hydrophobic overlap is maximized for this kind of structure when all the beta strands have the same amino acid sequence. But you might also notice that all the charges are also overlapping. In fact, overlapping of like charges was considered a reason why this structure was unlikely in the early days of amyloid science. So I'm going to talk mainly about an oligomer of Alzheimer's beta amyloid 42. It's an oligomer composed of 32 molecules. It is thioflavin negative. That's an amyloid sensing dye. It does not seed fibril formation, so this is not an amyloid fibril. And what I want to know is, why would a structure like this form beta sheets, reach a certain size, a relatively small size, and then stop? Why wouldn't it grow further? In fact, my intuition when people first started talking about oligomers is that oligomers don't really exist. You know, this was why I believed it was not, maybe they exist transiently, but how could a stable structure have this feature? I now believe that I'm wrong, and these are the people who primarily showed me I was wrong. So Danting Huang and Yuan Gao are two PhD students from my lab. This is a collaboration not only with my group, but also Terry Rosenberry at the Mayo Clinic, Hongchong Zhou at Illinois Chicago, Kong Gu at Shanghai University, and most recently Scott Stagg at FSU. So these are EM images taken from various papers. So amyloid fibrils, we spent a lot of time talking about in the last talk, but we know that non-amyloid structures also exist. So this is an EM image where you see what looks like nanoparticles. They're approximately spherical. This is an oligomer. Their structures are kind of in between that we call protofibrils. They're still much smaller than fibrils and slightly elongated. So what is an oligomer? So my working definition of an oligomer is any aggregate that is less than about 50 molecules, and they tend to be roughly spherical. There is some evidence that is widely recognized that the structures of oligomers may be distinct from fibrils, for example, antibody recognition. Some oligomers are thioflavin negative, not all. The beta strand organization has been proposed to differ. I'm going to talk about solid state NMR. I'm not going to get into details, but there's basically two types of experiments. Mostly, our 2D experiments are mostly carbon-carbon 2D, in which off-diagonal peaks tell you that atoms are near one another if they're isotopically labeled with carbon-13. And these are dipolar recoupling experiments where the decay, which we can simulate, indicates the three-dimensional organization of atoms. So first, a little bit about Alzheimer's disease. I am no expert on the disease, but it's associated with neurodegeneration And the pathological hallmark is the deposition of amyloid plaques, in which A-beta is a major component. I'm not going to talk about tau aggregation, but that is also considered very important. That's another protein. In the laboratory, if you start with a solution of monomers, there is a typical sigmoidal decay in the monomer concentration. Usually what you can measure is a sigmoidal rise in the fibril concentration. And oligomers are believed to exist at sort of intermediate timescales. They're not so easy to detect by thioflavin, but they can be observed, as I showed with THT. Now, fibrils have a seeding effect. So our ability to study fibrils benefits from this, because you can add fibril seeds to a solution, you can amplify it, purify it in terms of a single structure, and get beautiful data. Now, again, I'm not an expert on this, but many people believe that oligomers are more toxic than fibrils. In this case, this diagram illustrates that hyperactivation of neurons occurs because of oligomers and not fibrils. In the least, I think everyone can agree that fibrils are not necessary for pathology, that you can observe pathology without without fibrils. In this case, you can extract endogenous oligomers and then show that they have toxicity. And so let me say a little bit about fibril structure. This is not a talk about fibril structure, but I think you need to know some things, or we need to focus on a few things before we talk about departures from fibril structure. So the early analysis of fibril structure was focused on the orientation of beta sheets, or beta strands within beta sheets. So I showed the parallel in register refers to how well aligned the molecules are. The arrow refers to the N terminal to C terminal vector. So people wanted to know, do we have parallel or anti-parallel orientations? Remember that anti-parallel allowed you to separate the charges. And it also does not require fewer hydrogen bonds. So perhaps it's similar in energetics. And again, those who are familiar with fibril studies probably know that when it comes to fibrils, the in-register parallel structure wins most of the time. So most fibrils of proteins or peptides above a certain size are in-register parallel beta sheets. Not all. It is well known that not all of them are, but most are. And the NMR experiment that really showed this was dipolar recoupling. This is a pi-thirds experiment, the latest iteration of the technique. If you label at a single backbone site, a backbone carbonyl or alanine methyl, anywhere on the beta strand, then what you'll measure is a decay consistent with about a 5 angstrom distance. There are some systematic errors that exaggerate the measured distance, but you can see it consistently up and down the beta strand, and the interpretation is an in-register parallel beta sheet. Now, as Roland showed us, there's a lot of variations on that theme. So these are all fibril models. Some of these are very detailed structures. And you can see that you can have more than two, you can either have 2 or perhaps 3 or more beta sheets, and the way in which they stack differs, but all of these are in register parallel beta sheets. Now, this is every NMR spectroscopist's dream from the Griffin group, right? So a high resolution spectrum in which you can really get a structure out of it. This is the latest state of the art in data from an amyloid fibril, and we now have a high degree of confidence. Of course, NMR is not the only technique. Cryo-EM has also shown a great deal of information. So this is another in-register parallel beta sheet structure. Again, this is still a beta, and you can see that there's quite a bit of variability in where the beta strands are and how they organize. Now, this is an old measurement, Fourier Transform Ir. So this was a competitor to solid-state NMR in the early days. IR spectroscopists were pointing to this peak marked by the dashed line, and they were saying that amyloids were anti-parallel beta sheets. Now, what has eventually been understood, I think shown, is that, that this peak tells you that you have a beta sheet. And if you also see this peak, then you have an anti-parallel beta sheet. However, if you have structural heterogeneity in your sample, you can see this peak when you have both parallel and anti-parallel beta sheets. In most cases, if you can purify fibrils and get rid of oligomers, you get rid of that peak. And so I think it's fair to say that NMR won this competition, that NMR showed that we had in-register parallel beta sheets. But I want to say something. This peak is real. So we cannot ignore its existence. Anti-parallel beta sheets do exist in A-beta assemblies, and that's an important point. Okay, so I'm going to talk a little bit about isolating oligomer samples. That is a major challenge. And then I'm going to sort of get into our structural measurements, and I'm going to try to expand the scope and ask what structures are possible for A-beta oligomers. I'm not going to answer the question fully, but I'm going to share what I think are some interesting thoughts. So Terry Rosenberry's lab showed that if you take a monomeric A-beta 42 solution, this does not work with A-beta 40, and you introduce a moderate concentration of SDS. This is just below the critical micelle concentration, far lower than the concentration of SDS in SDS page. Then what you can do is suppress the sigmoidal diaflavin T curve. And however, this aggregate at moderate concentrations of SDS is also a beta sheet by circular dichroism. If you do SDS page, you'll see some dissociation, and you'll see this ladder of monomers, dimers, trimers, etc. And so you can isolate oligomers. It first forms a 2-to-4mer at high SDS. If you dialyze away the SDS, the 2-to-4mers undergo further assembly, but it stops at approximately 8 to 50 mers. We call it a 32-mer. That's a light scattering interpretation. And so this is a SEC-MALS data set, size exclusion in series with light scattering. It shows a clear oligomeric peak. Now, we also looked at what happens if you do a solid state NMR measurement and then resuspend. There is some effect, but we still see something that looks like a oligomer. And I want to point out, I'm not going to focus on this, but you can actually take EM samples, take EM images from aliquots of different portions of this SEC peak. And if you look at the high molecular weight end, you do see some elongated, or well, you just see a lot of elongated particles, right? So we believe there is some tendency of these particles to elongate, but not to sizes that approach fibrillar sizes. So to summarize, we make NMR samples by a series of size exclusion chromatography, SDS, and then dialysis. We started off with lyophilization to prevent further aggregation, but more recently we use ultracentrifugation to fill an NMR rotor. So this is another piece of evidence. So after a week of NMR scanning, we see some evidence of an increase of diaflavin, but not to the level that's typical of fibrils. And again, we see this SDS page ladder. So I think we're in a position to ask the following question: what limits this oligomer size? Why is it diaflavin? Why negative? Why doesn't it seed fibril formation? So let's get into some structural features. I first want to say that we are not looking at spectra like the best spectra of fibrils. These spectra have fundamental disorder to them. They look more like the early NMR spectra of fibrils. That may be a fundamental limitation. We are not sure. But we use selective labeling. So this is synthetic peptide, selective isotopic labeling, either 2D NMR or dipolar recoupling, molecular modeling, and spin simulations. You can see how much centrifugation helps us. So it's not easy to centrifuge these oligomers. It takes about 48 hours at 200,000 times G. But if you do centrifuge them, you can get an NMR signal without lyophilization, and the red contours show roughly the same peak positions, and some narrowing. These are still not as good as the best fibrillar spectra. So when we look at the 2D, we can look at the peak positions, the chemical shifts compared to data from the BioMagRes bank, and we can see what we call secondary chemical shifts. I don't want to get into it, but they identify 2 beta strand regions. Notice that our line widths are generally above 2 ppm, in some cases well above 2 ppm. So if we do the same dipolar recoupling measurement that showed that fibrils were in register parallel beta sheets, notice that regardless of where we label in a fibril, we see decays in this band between what would be 0.5 and 0.6 nanometers in our simulations. In the oligomer, looking at the C strand, we see much weaker decays. There is some decay, but we think that that's relaxation effects or maybe natural abundance background. We don't think these atoms are near each other. At every position except for one, the valine 36, and we interpret that to mean that the valine 36 is at the center of an anti-parallel beta sheet. Notice at the ends, we have I32 and V40. And so if we label those and do a 2D NMR experiment, we can readily see proximity. So we can verify that we have an anti-parallel beta sheet. We thought this meant that we have a fully anti-parallel beta sheet system. In other words, the other beta strand, the N-terminal beta strand, should also be anti-parallel. It turned out that that was wrong. So if you just pick a center, these arrows indicate an interatomic distance, which means that if we label, say, the L17 carbonyl, we should get a strong decay in our pi thirds. What we found effectively was that this anti-parallel beta sheet has no center. Wherever we label it, we see a weak decay. So that's because it's not an anti-parallel beta sheet, but let me also say that we like to use these contact maps. So the rows and columns correspond to specific residues. If I have a colored square, it means that an all atom model predicts that we should see 2D NMR contacts between the corresponding residues for the row and the column. And the circles mean that we attempted to do the experiment and we did not detect proximity between these residues. So the N strand was much more problematic than the C strand. So I want to show you another rather strange thing. If you look at how the N strand is arranged relative to the C strand, you'll see that F19 looks like it's in 2 places at once. Okay, so these stars mean that we detected proximity. So F19 is near alanine 30 and I31, but it's also, which is at the edge of the C strand, but it's also at the center. Seems kind of strange. So let me tell you what the answer is. It took quite a while to show this, but this diagram within the N strand actually corresponds to a registry shifted parallel beta sheet. And once we started looking for that, we saw plenty of 2D NMR contacts. It's more complicated than that. If you label it more than one place on the N strand, what you can actually show is that it's not, the registry shift is not in a single direction, it's more of a zigzag. And then if you, this was the main reason we implemented the ultracentrifugation, if you narrow the lines, you can see evidence of inequivalency. In other words, there actually are 2 inequivalent F19s. They're not in the same environment. And this is a contact map where the yellow or the orange contours are our current state of the model. So these stars, if it's red, it's from ultracentrifugation. But notice that the stars actually line up much better with our model. There are some contradictions here. For example, we should see this cross peak, but we didn't. We think ultracentrifugation will improve our sensitivity, but we feel like we're getting there. So the question here, by the way, is this is bizarre, right? I thought it was impossible. This is one of the limitations of NMR. It can give you hints, but it's not always easy to resolve those hints, right? So we have a parallel beta sheet in coexistence with an anti-parallel beta sheet. I would have told you that that was geometrically impossible. In fact, I did tell my students that, but I was wrong. So these are low resolution cryo-EM images. I'll show you a better version soon, but notice that it predicts something that looks like a tetramer with a hole in the middle. And if we take our model that I'll explain shortly, and we blur it out, we actually get something that comes close in symmetry and dimensions to the 2D cryo-EM reconstruction. And this is our current best attempt, right? So we're starting to get better resolution in the 2D reconstruction, and our model, we are optimizing it for agreement with this density. And let me explain how this structure works. So we don't have a single molecular conformation. So this is our model. And what I've done is color-coded half of the molecules blue or teal, half of them yellow. They're all A-beta-42. They're all the same molecule. But what I'm doing here is I'm just drawing only the yellows and only the blues, but they're in the same structure. And now I've circled the yellow, and you can see I've got this sort of U-shaped conformation in which the molecule folds onto itself. In blue, I have an L-shaped conformation that's domain swapped. And let's just start at the C or the N terminus. We're going up to down. The neighboring molecule is blue, up to down. That's a parallel beta sheet. Okay, here I'm going left to right. The neighbor is going right to left. So we have an anti-parallel beta sheet in the middle. I do not believe that this model is a perfect representation of reality. I don't think you have perfect alternation, but I think that this can explain our NMR data. And so this is, again, that model. We can refine it from a recent paper in solution NMR from the Karula group. This is a structure. I think they have very solid data. But they're mostly structured only in the C strand, which is very similar to the C strand we measure. There are some capping N strands where the molecules are in beta hairpin conformations. But there's also heterogeneity. So half the N strands are unstructured, half of them have a beta strand. This is a tetramer. This particular structure does not have any parallel beta sheets, but it does resemble our the center of our model, the C strand. So if I take this model and I add hairpins capping, then I actually get better agreement. Again, still not perfect, but I think we are making progress. And I want to make I want to step back and ask, just make some general comments like that, explain things that I think we've learned here. And so going back to these questions, okay, so what limits oligomer size? So normally fibrils grow by monomer addition. I don't think this structure can grow by monomer addition. Okay, so yes, it's true. My original reasoning was correct in that you have dangling hydrogen bond donors and acceptors at the ends of the beta sheets. That is true. But in order for this structure to work, you need this perfect alternation or near perfect alternation of those U-shaped and L-shaped conformations. I think if you add monomers to this particle, then it's likely that you'll have errors, right? You'll have U's where there should be L's, right? And if you have too many errors, I think that is going to bring a stop to the assembly. In fact, I don't even think that a structure like this would form by monomer addition. I think that it may come from a tetramer structure, for example, and the tetramer could associate with other tetramers to form a structure like this. But that is speculation, of course. So why is it diaflavin negative? I think it is because you've got that weird zigzag pattern in the beta sheet that's on the surface. To my knowledge, a structure like that has not been observed. And I do want to comment that there are fibrils of A beta that are known to only weakly bind thiaflavin. So thiaflavin is not a perfect detector of beta sheet structures. Why does it not seed fibril formation? So I mentioned why I don't think it will grow along the beta sheet axis. I think the surface is probably somewhat disordered. And I think that weird zigzag structure probably is not very good at secondary nucleation. So I want to come back to this question of what is the difference between an oligomer, protofibril, and a fibril? Okay, I'm not going to answer this question, but I'll give you a few ideas. Okay, so now this is a very simple diagram. So most data on A-beta aggregation indicates that you have two beta strand regions. There can be more than two, but I think my idea here does not require more than two beta strand regions. So I've labeled what we call the N strand in blue and the red strand in the C strand in red. So imagine I have 4 monomers, and I'm just labeling the red and blue domains, but they would be random coil at this stage. Now, if for some reason the C strands came together and formed an in-register parallel beta sheet, well, I think you would agree that the N strands would be well oriented and can readily assemble into the same configuration. And so you would get an amyloid fiber, right? If you did the same thing, you assembled 4 N strands, then the C strands could readily adopt a similar configuration. And so you can get at an in-register parallel beta sheet by any region of the peptide forming an in-register parallel beta sheet. The rest of the molecule can just zip together. Okay, now what happens if that doesn't happen? So you could form a beta hairpin, so you have hydrogen bonding within the molecule. And Doi and Takagoshi have suggested that this is what happens in protofibrils. I don't know, I mean, I don't think that's proven, but it's an interesting suggestion. I think that this is probably a less efficient mechanism for A-beta. Now, what if you form an anti-parallel beta sheet first? Okay, so I'm going to go down this step. So an anti-parallel beta sheet by the C strand. And now Chang and Tico showed that you can also organize the N strand into an anti-parallel beta sheet in the Iowa mutant of A-beta 40. So they have observed this structure. I would call it a protofibril, but they called it a fibril. The terminology is not obvious. But I ask you this question, is that the most likely thing to happen? And I suggest that the answer is no, okay? I think it's actually more likely that despite you having an anti-parallel C sheet, the blue strands are actually still more likely to form parallel beta sheets because their nearest neighbors are oriented in the same direction. And this structure is a frustrated structure, right? So I don't think you can indefinitely add monomers to a structure like this because I don't think it can exist in a plane, right? And so, by the way, how is it possible that this could happen? Notice that sequence is very important. Remember, the C strand is more hydrophobic than the N strand. So remember, Rosenberry's protocol uses SDS near its critical micelle concentration. So that hydrophobic interface, I think, is what actually makes this happen. And it's also been shown that you can form oligomers using lipids or other detergents. I believe the Karula group used DPC. So the other comment I want to make is that almost every experimental report on an A-beta aggregate actually fits within this picture. Right, sure, I showed you fibrils with more than 2 beta strands, but I think you can use very similar reasoning to argue about those. The one configuration here for which I do not know of any experimental evidence is this one. So nobody to date has reported an anti-parallel N strand and a parallel C strand. Now, is that because it's not a reasonable structure or because nobody has isolated it? I don't know the answer to that question. And I'm done with my talk, but I do want to point out, so yesterday was Halloween. So at the end of a lot of horror movies, the monster comes rearing back, right? So this beast has not been vanquished. And so I think that we do not understand all the structures that are possible for A-beta aggregates. We do understand that it is complicated. I think some A beta scientists are under the impression that there's some infinite number of possibilities. I don't think it's an infinite number. It may not even be much bigger than the number that we currently know. But I still think that there are important things that we do not understand. And with that, I thank you for your attention and would be happy to answer questions. Thank you.
Great talk, and then nice structures. So let me start asking the first question here. In your sample preparation, do you have monomers and small oligomers together with your large oligomer, or you purify to only have only large oligomers?
It's important to separate the monomers away. Without doing size exclusion, you would have both monomers and two-deformers, and perhaps the monomers would persist if assembly went further. But remember that fibrils are better at forming than oligomers, right? So you've got to get rid of monomers in order to prevent fibrils from happening and isolate the oligomers.
So when you did the ultracentrifugation, what do you think is happening? Because you see the NMR spectrum is getting narrower, which means that you're narrowing down the structural heterogeneity. Is that what the interpretation?
That's possible, but I think a bigger effect is just that lyophilization broadens lines. So even if you have a very high homogeneity fibril sample, if you lyophilize it, you'll see significant broadening of the lines. And that's just because lyophilization is kind of a harsh treatment that will introduce disorder.
But here you do have water, right? The centrifuged.
Yes, so we're doing NMR of the centrifuge pellet, so there's a fair amount of water present.
Okay, now I take the questions from Q&A folder. If Miller is asking, oligomers are supposed to present polymorphism. Can you follow this via NMR? That's the first question.
In principle, yes. In practice, that is very difficult. So I do think polymorphism is perhaps even more likely for oligomers than fibrils. So an oligomer is not one structure. I'll be very surprised if there is only one oligomeric structure. But NMR requires that you produce a sample that is structurally homogeneous and stable. And I think I want to add that for some oligomeric structures, that may be impossible. I think this structure may be special because it is actually an endpoint structure, right? It does not undergo further aggregation. Some people would call it an off-pathway oligomer.
Okay. Are all A beta oligomers form beta sheets? Small oligomers are not expected to form well-organized beta sheets.
So I think that is probably true. And I believe that there is evidence, for example, of alpha helical structures, especially at very high levels of STS. And now the term oligomer means different things in different contexts. Some people would call a liquid-liquid droplet an oligomer. So that is not a beta sheet. Some people believe that fibrils are nucleated out of liquid-liquid droplets. I'm not sure I answered your question.
Yeah, you mentioned about being off pathway, this oligomer. So does that mean it's a toxic or not at all toxic? Did you test it?
That's a good question. So this is known as the globular. It actually was not invented by us or Rosenberry. And there are papers that show some evidence of toxicity. My own personal opinion, and this is not a rigorous thing to say, is I think the two-deformers are much more likely to be biologically relevant than the 150 kilodalton structure. And so let me tell you my pet theory. It's not my theory. In fact, I read it in a paper by Wolf. And what he argued was that when APP cleaves A beta, some of it stays in the neuronal membrane. And in the neuronal membrane, you have something that resembles an SDS micelle, or maybe that my SDS resembles a membrane. And so you could be forming two-deformer structures in the neuronal membrane, and it may never leave the neuronal membrane. Now, what we are seeing is what happens when you take the A-beta out of the lipids. I think it tells us something about that two to former structure. But if I had to guess what is the most important oligomeric structure, I don't think that the 150 kilodalton structure is it. But that's not our purpose here. Our purpose is to understand what is possible.
Okay. So Jeff Brender is asking, do you think alpha helical intermediates, for example, those induced by HFIP, can be fit into the 1-to-5 scheme shown in the last slide?
So yes, in fact, we have other work. It's on YouTube. On a designer system in which you have alpha helical assembly, and then that is metastable, and the helices will actually uncoil and form beta sheets. This is not work on A-beta. It's on the SAF system by the Wolfson lab. But I think that is evidence of what you're suggesting. So I think that alpha helices can form oligomers, and that those oligomers can convert into beta sheets. And so it doesn't exactly fit into the scheme, but it is perhaps possible to use an alpha helical structure to create a certain orientation of beta sheets and then uncoil the alpha helices in order to form a beta sheet oligomer that is in the scheme. Very speculative.
A question from anonymous attendee. In your publications, you reported preparing 150 kilodalton oligomers by adding SDS. How homogeneous the preparations are?
So in terms of biophysical measurements, they are pretty homogeneous, right? So light scattering gives you a single size. The SDS page gives you some evidence of dissociation, but a fairly well-defined size. The NMR line widths are not as good as the best line widths of fibrils, but we think that they do define a specific structure. We have also analyzed our dipolar recoupling decays to ask, could you explain the data with a certain amount of parallel beta sheets coexisting with anti-parallel beta sheets? We've seen that with our work on designer peptides, but we do not think we can explain the data on A-beta that way. So I do believe that it is a structurally homogeneous but fundamentally disordered structure. I know that sounds like a non-answer, but it's the best I can give you.
Another question from anonymous attendee that, where is it? It seems to me regarding to oligomer model you showed, there's a lot of large space in center of the oligomer that might allow water molecules to be involved. Is this correct?
Yes. So in fact, the Karula group showed that this forms a pore. So they did electrophysiology measurements or measured current. I shouldn't use big words. So probably got it wrong. But yes, they have measured current through the membrane with this oligomer in it. We have not done that measurement, but based on just the size of the pore, we do believe it is big enough for water to go through. And in fact, that's probably even true in a few other areas, not outside of the center.
The next question from Ada Caponito. Thank you for your nice presentation. Did you try to generate an antibody against this oligomer to test whether you can find it in the AD brain? Does it bind A11?
It does not. Oh, I'm sorry. It does not bind A11. Let me go backwards. Others have made globular specific antibodies. And we have not personally done brain studies, but I believe that others have done brain studies. One thing I have to be a little careful of is that even though our prep is based on the globular prep, we modified it for compatibility with solid state NMR. So we are not 100% sure that the structure that we studied is exactly the same as the structure for which antibodies have been raised. But I do think that is a very important line of inquiry. I think it's probably the only way to find specific oligomer structures in the brain.
Another question in the Q&A folder, Romani is the person. Great talk. Are you do shaking for the oligomers in the presence of SDS?
sorry, there was a choppy sound. What's the
Okay.
beginning of the sentence?
Or you do shaking for the oligomers in the presence of
Oh.
SPS.
Shaking, not that I know of. Rosenberry's lab does that, and I don't believe shaking is involved.
Okay. There's in the chat box, couple of questions. Let me address, take that. Great talk. How many oligomer species in the sample and what is the pH of the NMR experiment? Does the oligomer stable at low pH? Is this for you or for Roland? I think this must be for Roland, I think. Okay, let's see.
Well, Roland did measurements along those lines, so we have
Yeah.
not investigated the stability at different pHs of the, at least not that I know of.
Okay, there's one question.
But I didn't talk about oligomers. I think it's
I think your beginning portion, the granule amyloid protein stored in the granule pH as being acidic. So we can come back to that. Let's take the question from Carmelo La Rosa. TERS measurements, simultaneous Raman and AFM showed that IAPP and A-beta 1 to 40 fibrils are not homogeneous. Beta sheet and alpha helical structures are observed. Is it possible that NMR measurements lack the resolution to observe this inhomogeneity in fibril structure?
So I don't think the right lack of resolution is the right way to put it. It is, I think the way I would put it is that if you had fibrils that had a fundamental inhomogeneity to them, you know, the presence of multiple distinct conformations, it would add complexity to the NMR spectra, and it would cost us in terms of resolution. So we measure those all the time, right? So it's not hard for us to get bad NMR spectra, right? But exactly what the interpretation is not easy, right? For this reason, there is a tendency among myself and other NMR people to try to study the samples and give the best spectra, right? So if you get nice sharp lines, people like me love them, right? And so I think in nature, I don't think nature is obligated to produce aggregates that are highly homogeneous. And so this is a bias. It's a disconnect between our scientific methods and what may occur in nature. So so the answer is not that NMR cannot study these things. It's just that, well, it's not easy.
Well, can we rule out alpha helical structures in your sample that is inside the NMR tube? So that's, I think, yes.
Yes.
Okay.
Yes. So in this case, yes. But that doesn't mean that aggregates containing alpha helical structures do not exist.
Correct.
That means that we're doing studies on a prep that doesn't produce that structure.
So, so it's off pathway and less toxic, or maybe that's what you said, right? Your oligomer is less toxic.
I don't know that it's less toxic. I
Right.
speculated that it might be. I don't know that.
So it's free from monomers and small oligomers. How stable is it? So if you take the oligomer and leave it outside in room temperature versus low temperature, can you give us some idea about the stability?
So it's stable for weeks.
And what happens to that?
Eventually, if you leave it in solution, eventually you'll get fibrils.
Oh, so it doesn't go back to monomers. I see.
So, well, I think it is necessary. So this is my working definition of off pathway, right? So an off pathway oligomer needs to dissociate into monomers in order for fibrils to form, right? So remember that the assembly is reversible, right? So if you, I believe that everything is reversible to some degree, right? And so if we are forming a structure like this, my definition of an off-pathway oligomer is that it has to repopulate this state before you can go down a fibrillar pathway. And so it is not true that an off-pathway oligomer isn't transient, right? In fact, it is transient. And in fact, if you put any oligomer, well, okay, I don't know this, but I think that if you put any oligomer in competition with a fibril, the fibril will win.
So there's another question from chat box. I think it should be for you. Do you have HD exchange that shows that the oligomer is well structured and not only like a micelle-like entity?
HD exchange?
Yeah.
To my knowledge, HD exchange has not been applied to globulomers. We have done some measurements using hydroxyl radical footprinting, which does measure solvent accessibility. And the oligomer showed some evidence of protection from water, but it was not as good as fibrils, and it was not. It was also clearly not monomeric. So we have not done HD exchange, to my knowledge, and I believe the NMR spectra indicate order, right? So that there is a homogeneous structure with some degree of heterogeneity. It's not a completely random structure.
Yeah.
And that is clear from the NMR spectra.
So there's one question Q&A folder by Rakesh Kayad. Great talks, both of you. Question is for Anant. Any idea about hydrophobicity of these oligomers and fibrils versus THT?
I don't have a good answer for you, Rakesh. That's a good question. So you're suggesting that that this does not present a hydrophobic interface that can bind to thioflavin T?
That's what looks like, yeah. Yeah, okay. Rakesh is raising his hand. I'm going to allow him to directly ask the question. So, and he also says that these are toxic oligomers. That's what he thinks. Let me just see. Okay, as you are in the panel, you can go ahead and ask the question directly. Okay. Just a second.
So my comment is about the oligomers. So if they are thioflavin T negative or weak, normally they have hydrophobicity and they are very highly toxic. So I agree with Anand's assumption that these are toxic.
Sorry, can you say it again, Rakesh? If they don't bind
I
to
said
THT.
Anand oligomers were weak with thioflavin T, if I understood correctly.
Yes, correct.
These are normally the ones which have higher hydrophobicity or BCNS, and they are more toxic than the ones which have high thioflavin T signal. We repeated these experiments zillion times, so they're the same.
Oh. So is it possible that high thioflavin binding might also indicate an on-pathway oligomer, which may mean that their concentrations effectively reduced by fibril formation?
Yes, and I think there is a paper, I'm not a co-author, but from Charlie's lab, long time ago, a few years ago, comparing both of them, the ones which are on pathway, off pathway. I think this is, I would say this is an accurate statement. Where is the sweet spot between beta sheet and hydrophobicity? I think this is an important area of investigation. Great talks, both.
Yeah, thank you very much, Anand. So now the session is open for questions for both the speakers. Please feel free to raise your hand or post your questions in the Q&A folder. We can address them. So Anand, going back to your samples, let's say, did you ever try without purification? So you will have a mixture of monomers, small oligomer, large oligomers, and stick it in the NMR tube, or maybe can you find out the size distribution in your chromatogram to see if you have coexistence of other small oligomers, and we can do NMR as well.
Yeah, so you can resolve in size exclusion. Let me see if I can go back to that slide. You can resolve oligomers of different size as well as monomers in size exclusion. And I think that, so I should also emphasize that Terry Rosenberry's lab does these measurements, and so I have no firsthand knowledge, but I've talked to him often, and so this, I believe, is the monomer peak. Sorry, I have to think. It's later eluting, so lower molecular weight. And you can see, so if you put oligomers through size exclusion, you will see distributions of sizes. And you can see monomers, but you can also isolate a specific size. So you can isolate this peak with the fraction collector and greatly reduce the detectable monomer if you run it through again, through the column again. But I don't know that you can completely eliminate the monomer concentration. And with NMR, we're skittish to do experiments like what you suggest, because in fact, for a very long time, we kept our samples completely dry, right? We lyophilize and made sure there's absolutely no water, which we can measure in the proton NMR signal. And that was because we were worried about fibril formation. So we did see some evidence of it. So you can see some increase in thioflavin. So you can even see it at this slide. So when you resuspend, the thioflavin does increase, not nearly as much as what you would see in a fibrillar sample, but we were very concerned about the stability of our sample in the NMR measurement. Maybe now that we have more understanding, we can be a little more adventurous with our sample prep, but we have avoided that kind of experiment because the structure changes. And by the way, if you put a monomer, if you lyophilize monomeric A beta and try to do NMR, even in a lyophilized sample, it will assemble into fibrils during the NMR measurement.
Yeah. So what is the yield of this large oligomer? If we take, let's say, X micromolar concentration monomers, you prepare and then you get your 150 kilodalton oligomer. How much is the yield? Is it 50%?
It's probably less than 50%. I don't remember exactly. It really depends strongly on the source of A beta, right? So the microwave synthesizers, the CEM Liberty Blue, gets the best results in our experience. And if you treat A beta right, the first step requires separating monomers from aggregates. So even in freshly synthetic A beta, you can often get a lot of aggregates. In the best cases, we approach 50%. But you have to be very careful. You should, Rosenberry has told me that it's not good enough to assume that a solution is monomeric just because you've used HFIP, DMSO, or high pH. You should run it through an SEC and isolate the monomeric peak. And you could lose a lot of peptide in the process.
Thank you very much.
So it's very tricky.
Yes. Roland, I have a couple of questions for you.
Of course.
In your PRE experiment, you showed the NMR data. Wherever you show the intensity, there seems to be an oscillation pattern in the N-terminus. Is it right? Do you have a structure then?
Yeah, it is. I mean, Synuclein has these repetitive elements, but there's every 7 residues. So it seems to be that there is some specificity to certain amino acid types. But we didn't look into it more detailed.
Is it that, or do you have some kind of local structure which is periodic, and then the PRE effect could be based on the structure? Don't you think so?
It could also be that, yeah. We didn't look into more details, but that's an interesting observation, yes.
And what about your in-cell NMR? How much is the synuclein that is going inside and then as opposed to being lost on the way?
Yeah, so we have about a few micromolar synuclein we can put in. We can do a lot of proteins in, so usually in the low micromolar range, like ubiquitin or PDC domains or cyclophilin or enzymes. So they give okay spectra or other IDPs like tau or protein alpha. So that works okay. It's a lot of protein you need to put, and it's not so easy. You cannot do so many experiments, but it's just cell biology is.
Mm-hmm. And do you think you're seeing all the protein that goes inside the cell, or some of them are lost as oligomers and fibers? which are invisible.
So yeah, of course we do solution state NMR. So that means we cannot define how much do we really see. I mean, how much do we lose? We did a functional assay. So we had a protein that activates transcription factor activation, and then we think that when we electroporate, that protein is at least 90% active. So it's not something that happens on the way. Interesting observations we have when we take familial mutants of synuclein and electroporate them, they get immediately cleaved. So there is activity of the cell, they don't like it then. I mean, you can still see them, but the C-terminal is getting cleaved away. So that's what you want, right? You want to see activities. Yeah. What we also see is when we add ubiquitin or PDC domains, we see also with synuclein, but with minor extent, we see multiplicity of peaks. So we see several states of the protein. That's probably transient interaction with other proteins. So that's what we now at the moment look into.
Okay, great. I think we answered all the questions. There are no other questions.
I have one simple question. So to both of you, for NMR studies, are we or somebody looking at brain-derived oligomers for A-beta, synuclein? Because, you know, the cryo people are really doing good job, but still it's the core. We need more NMR studies. So
I think Raptico is working on brain-derived A-beta oligomers.
So the problem is, maybe to summarize a little bit more extensive. So with NMR, we need large quantities, and cryo need very little. So what people usually do is use like PMCA. You try to multiply with synthetic material the conformation. And at the moment, it looks like there is this disagreement between the cryo-EM structures to a certain level and the ones that are amplified. And there is also not known what is actually true, because the cryo-EM structures use sarcosyl as a detergent to extract. So you might induce certain conformation, but the recent Nature paper by Sheres showing for 9 tauopathies, different tau structures, indicates that's not the problem, at least in tau. But in the synuclein case, it's more open. I mean, it could be also a purification artifact. And what I understand is that Michel Göder, they try now to do antibody pull-down experiments and other ways. Or the PMCA may not work because you don't have the right components to amplify. You may need an RNA. I mean, we know in synuclein fibril structure prepared from MSA patients, there is some molecule in there which we don't know the origin of. But whatever we do, what is very important, I think, is that we are able in vitro to replicate the in vivo structures, because we need that to understand and to do much more intensive research. So we need to come from both sides. We need the in vivo and the in vitro approach to understand finally these polymorphisms.
I couldn't agree more. And I guess the one thing I would add is that this is why we want to know what structures are possible. It's not just a question of what you have in the brain, which is certainly an important question. But if we can understand aggregation in holistic terms, you know, what structures are possible, what structures can convert into other structures, how does the assembly work in a comprehensive way? That's a very hard question, but I think a very important one.
There may be a last statement on this. Is also Charles Weissman showed that in prions you have different polymorphisms in the same sample, but some of them replicate faster in a certain environment, another one in another environment. And so it may also be that we purify one strain out of the system in this procedure and another one in another procedure, and that at the end there are multiple polymorphs, and in principle there need to be multiple polymorphs. Just from Boltzmann argumentation, it just matters how many of them, which one you have.
We'll take one the
Thank
very
you.
last question for today. Anand, it's for you from Max Lindbergh. Have you considered using recombinant A-beta?
Yes. So we have avoided it because our line widths are rather high. So we are in the sort of 2 to 4 ppm range. But recently, Bo Chen's lab has been getting good NMR spectra, or actually the line widths are not more narrow, but they're able to make assignments, which I find very impressive. So I do think that uniformly labeled biosynthetic A-beta could be studied in a meaningful way.
Sorry, why did you get broad lines? Because of dipolar couplings or what?
Because it's oligomers. So there is some
But you
fundamental
said
heterogeneity.
your biologically expressed one gave broader lines than the synthetic one. Is
Oh,
that right?
no. It's so in fibrils, people use biosynthetic A-beta to get uniform labels, right?
Yeah.
And that is very specifically for the preparations that give very sharp lines, sub ppm. So you can go to high magnetic field. Oligomer samples, to my knowledge, nobody has come up with a prep that can give you those sharp lines. And so we've always used selective labeling to simplify the NMR spectrum. But I do think that that this is something that we should do.
Thank you very much. Thanks to the speakers, and thanks Sebastian Hiller for making this more eventful. And thanks to all the participants. And thank you all very much.
Thank you
Sure,
very
thanks.
much.
Bye.
Bye-bye.
Take care.